Good afternoon. Welcome, faculty, staff, students, and community members to the Science and Faith event. My name is Louise Huang. I direct the Center for Research in Science, and I am so glad that you can come and learn a very important topic from a very special guest speaker today. Dr. Ting Wu received both her bachelor and doctorate degrees from Harvard University. She then somehow migrated to the great state of California for her postdoctoral research at Stanford University. But seemingly, her New England roots beckoned her to return to her alma mater, where she pursued a career as a genetics professor at Harvard Medical School for the next 26 years. She is a very accomplished researcher with numerous publications and a thriving research lab that studies chromosomes. She is also the recipient of 3 National Institute of Health awards, including the Transformative Research Award, the Pioneer Award, and the James A. Shannon Award. She's a well-sought-after speaker who appears on TED Talk, major conferences and forums. But that's not it. Dr. Wu is also the founding director of the Personal Genetics Education Project, also known as PG Ed, through which she would lead congressional briefings and teach genetics to the younger and the older, from Boston to San Francisco to Baltimore to our neighbors in Boyle Heights. According to the renowned Harvard geneticist, Dr. George Church, she is a remarkable researcher and a gifted teacher. So would you help me warmly welcome, for the very first time at the CCCU, Dr. Ting Wu.
That was really so very generous of you. Thank you. It is really an honor to be here. The second I got Louise's email, I knew I had to come out here. And you'll learn about this slowly as I go through my talk. I've really had a wonderful day. And I'm here very, very much, in part, to hear what you have to think, to say, to comment on in this field of genetics. It is absolutely essential that geneticists understand how the world sees what they do. And that really is why I'm here. And thank you very much for inviting me. I've had a wonderful time already. So now I seem to be coming, I think I'm recovering from a cold. If you can't hear me, raise your hand, and I really apologize for the sound of my voice. So, okay. You can't hear me, so I'm going to pull this out, I think. Does this sound better? Wow, okay. So I basically said thank you for having me here, and the most important part is I would really like to hear your thoughts. So if we can, I'm watching the clock, we're going to stop at 5:15. I don't have to get through every single topic I have. Stop me if you have something to ask or comment to make, because in the end, what we really need to do is have a conversation about genetics. So I thought that I would introduce myself by introducing to you the students and the postdocs that I work with in the research lab. As Louise said, we study chromosomes. We have invented a technology for looking at chromosomes at a resolution really somewhat unprecedented. They are beautiful to look at 20 nanometer resolution. We play a lot together, we celebrate together, and a few years ago, Mohammed got his citizenship, so we celebrated his citizenship together. We also sew our Halloween costumes together. This is my lab. We did not win the prize, and we also go out and play together. But what I want to point out is that there was a time when I was the only US citizen in the lab. People come from all over the world. And so in our lunchroom, when we talk, we debate all kinds of things, from politics to women's rights, to racial issues, to what is healthy to eat and what is not healthy to eat. And most importantly, we talk about where genetics is going. Because these young people, just like all of you, are going to have a completely different life, different from any other life that a human being has lived, in a way so dramatic that they are grappling with what they're going to have before them when they decide to have their children. And so about 12 years ago, as Louise mentioned, Dana Waring and I founded the Personal Genetics Education Project. It sounds so formal, but it really was just the two of us thinking we needed to hear what the community was thinking. Not so much go out and, I actually have issues with the word education, but that somehow got into our name, to educate ourselves. And our goal is actually not to promote genetics or to critique it. Our goal really is to raise, to promote awareness and dialogue, and through that respect. So people sometimes find it odd that as a geneticist I'm not out there promoting it. I really am not. We are at the crux of a very complicated time, and I will say that geneticists have not listened enough to what the general public is thinking. And so this is just one effort among many geneticists. Many of my colleagues are doing similar things to raise awareness, dialogue, and respect. Here's my team. They do all kinds of things. We recently hired a set that is bound, their goal is to make the Boston public the most knowledgeable about genetic technologies as sort of a pilot program. And I want to tell you that we were supported in the beginning just by scraps from my lab. But then one day my chair, Cliff Tabin, he's the chair of my department of genetics, he came to me. I thought he was going to tell me that I was spending research money inappropriately and that I would have to shut down PGED, but instead he came to me and he said that he felt, as the chair of the Department of Genetics, he had a moral obligation to reach out to the community that we serve. Because in our department are some inventors, including George Church, who invent some of the technologies that are really accelerating genetic studies. And he realized he could not, in his heart, feel good about supporting that with his right hand, with his left hand, making sure that he was talking with the general public. So I just want to thank him very much and let you know that I really represent quite a team. And it's an honor to work with them. All right, so I'm going to briefly tell you that one of the things we're shocked by is the speed with which genetic technologies are changing our lives. Really, it's just a little over 100 years ago. Now, for those of you who are young, 100 years seems like a long time. But for those of us who are older, 100 years is so little time. And when I think about the fact that in my laboratory, sometimes it takes 2 years to get one experiment to work, I cannot believe the pace at which genetic technologies are rising. So this is Nettie Stevens, who is my personal hero. In 1905, she was the one who discovered the Y chromosome, and for the first time said, back then they did not know chromosomes were the genetic material. She's the one who said, I think I've connected a phenotype, maleness and femaleness, to this thing that we call the chromosome. And it's because of her that modern genetics was launched. Now recently, she has enjoyed an increase in popularity because Google celebrated her birthday. And these are some of the drawings that her colleague, Charles Metz, drew for these chromosomes that they were trying to understand. She died very young of breast cancer, but her legend lives on. Now, at that time when they discovered that something can be inherited, now before that, they were unsure that heritable material was even a substance that could be passed on. But the second they found these chromosomes and connected traits to those chromosomes, the world became enamored with the idea, and we're going to come back to this at the end with a little twist on this theme if we have time, that genetics is based on a substance. And that substance is passed along. This is a typical kind of pedigree that we have. Circle for female, squares for males, diamonds for gender ambiguous. And we have now wonderful symbols for adoptions, for surrogates. Even these symbols themselves tell us how rich our genetics is. So that is where we were. And then human beings started inventing things. I was writing the plane here, just sort of floored, and I feel floored frequently at Homo sapiens. We are such an extraordinary species. We do things that are stunning and shocking because we complain a lot. We complain about this and we invent something, we fix it. We complain about that, we invent something, we fix it. The world is too small, so we invented and got onto the moon. But these inventions are dramatic changes in our lifestyle. So in 1969, we left Earth and we landed on another celestial body. Less than 10 years after that, we took another gigantic leap, that we started life in a petri plate. Stunning. You wouldn't have guessed back at the turn of the century that you would be able to do that, but now millions of children have been born, starting life in a petri plate. So here's Louise Brown, the first test tube baby, and there was tremendous joy across the world that she was able to be born. There was also tremendous anger and angst and threats that a test tube baby had been born. But Homo sapiens just keep moving forward. And then we came to less than 10 years after that, we hit the genome sequencing era. So I was a graduate student at that time, and I will tell you I need to tell you my conflict of interest, but I have no conflict. I'm married to George Church, who was one of the people responsible for the genome sequencing era. And I remember when we were graduate students talking about this, it seemed impossible, but yet it seemed so inevitable that we would, as human beings, want to see our genetic material. It took 13 years, $3 billion. We got one sequence. It really wasn't even a pure sequence. Today, in 3 days, for less than $1,000, you can get your genome sequence. Pretty soon, the idea is that you'll be able to get your genome sequence in hours, and to make it accessible to everybody who wants to have it, the goal is to make it essentially free. Maybe part of your healthcare system. You go to the doctors, they draw your blood, you get your genome sequence, if you want it. This is one of the most modern genome sequencing machines. It's this little device that you can stick onto your iPhone. They're inventing it now. You take a sample, you pull out a little drawer, you put the sample on it, it sequences it, it reads the information into your cell phone. They're still working on it. The current version that's out is just a little bit bigger than that. It's about the size of the cell phone itself. So this is the world in which all of you are going, all you young people are going to be raising your families. It's the world in which we are now. So this pedigree, where you would have to learn about your history by making sure you understood who your grandparents were and what their health issues were. Now it's very different. You can learn a lot about yourself and your genetic inheritance without knowing who your parents are. And this is extremely important for children who are adopted. In fact, adopted children are often the first ones in a group to want to be sequenced, because that is the way they come to know their ancestors. And you can get blood from get DNA from saliva, cheek swabs, blood, hair roots, tissues, and get information. Now, I could pick a variety of traits to talk about as to how people are using this, but I want to talk about this one, although it is not medically as actionable as we would like it to be. And that's because this is one that I personally feel is important for everybody to know about. So CYP2D6 is an enzyme. And what this enzyme does is it metabolizes codeine into morphine. There are 100 known variants, and they keep finding more. Why am I talking about this? Because there's some of you who are normal metabolizers, and some of you very poor metabolizers. My daughter is a poor metabolizer, and we discovered this when she was having dental surgery and the codeine was not working. If you are a poor metabolizer, and a good 10 to 20% of you will be poor metabolizers in this room, you need a little bit more anesthetics for various kinds of surgery. But the dangerous one is if you're a rapid metabolizer. So I don't know if you've ever gone to the doctor says, are you allergic to codeine? In the future, geneticists and physicians hope that you will already know because you have been tested for this. Because if you are a rapid metabolizer, you will metabolize that codeine so quickly into morphine that you could be in danger of dying. Now, the way people figure this out, whether they're a rapid metabolizer or not, is because they have a reaction. But fortunately, hopefully, they're sitting in a hospital and the hospital can revive them. In some communities, in some ethnic groups, rapid metabolizers will be up to 30–40% of everybody in that group. So I mentioned this to say this is one of the reasons why personalized medicine is moving ahead, because if we can let people know at the time they're born what they're at risk for, we may be able to minimize the amount of adding. Now, this is a difficult one to look at, but I have many colleagues who are working very hard at this one. Actually, some of you may not know, but as soon as you're born, 20 to 30 to 40 genetic tests are done on you to help your parents, prepare you, for example, if you have phenylketonuria, to help you with your diet. And this was one that I would personally love to see on that panel. Okay, now we hit 1990. 1990 was a time when people who were doing in vitro fertilization realized with all these genetic technologies for looking at sequence, they could actually look at an embryo in a petri plate and learn about its genetic makeup. This is an 8-cell human embryo, 8-cell human embryo, where the researcher, physician, has taken out one cell. That one cell can now be genetically tested, not so far to get a whole human genome sequence, but can be tested for a variety of traits. This really was another milestone we took, because it enabled pre-implantation genetic diagnosis for gender-associated diseases, what it was first used for. And the reason we're here today is to talk about genetic editing. Genetic editing, a little bit later, 2013, about 13 years later, changed everything. But you'll see it actually, although it seemed to change everything, it really is just one of a whole series of advances that have been made to allow human beings to think about and possibly implement changing their own genome. So basically, we went from genome reading to genome editing in a very few set of years. And now I think the arrow is ethical considerations of genetic technologies. That's where we are now. That's why we're in this field. All right, so I told Louise that I would summarize genome editing, and I will tell you the scores, the hundreds, thousands of researchers that have made this possible. I will not be able to mention all their names, but they are really, truly brilliant people who, for many of them, the goal was to address genetic disease, to give options. All right, so in the beginning, people took advantage of viruses. Viruses carry DNA, and so what researchers did was to take a gene that they thought they might want to insert into a plant, for example, or a cow, possibly in the future, a human, and they would put that piece of DNA into the virus DNA, and these are a set of viruses that are often used, and then let the virus do what it normally does, which is to insert itself into genomes. It's kind of a gene transformation, a genetic transformation. The trouble is viruses go in all different places. You cannot target it to a single place. And therefore, when you use viral-based gene transformation technologies, you run the risk that the virus is going to sit into a gene that is one you do not want disrupted. And so, although people were excited about this ability to change genomes, they realized, one, they had the problem of random insertion, and then the second problem, which is very, very low efficiency. If you have a car and it runs beautifully, you love it, but it only starts 1 out of every 10,000 times. You're not going to want to use that car. And this was a huge problem. So then, so I should tell you that we went back to basic research. I know many of you do basic research, and learned that genomes have a tremendous ability to heal themselves. And researchers thought, is there a way we can take advantage of this endogenous capacity to heal itself? So for example, as you grow, you go outside and sit in the sun too much. You drink too much alcohol. your body will respond to the environment sometimes. Well, I shouldn't say respond. Sometimes your DNA will break. Well, the reason we're all able to sit here and be generally healthy is you have in you a tremendous, complicated machinery that recognizes that break, brings in another piece of DNA, makes sure it's the same kind of sequence, and then inserts that other piece of sequence into the break and heal itself. You do this hundreds and thousands of times. When you replicate your DNA, replication is very error-prone, and your DNA repairs itself. So, it wasn't a big leap for someone to say, wait. So if there is an error, and we're going to have to think about that word error a little bit, maybe we'll talk about it during the discussion panel. But if there's a change that we don't want, maybe what we can do is somehow cause a break to happen there. And then let the cell just do what it normally does and heal itself. So that in the end, you've pretty much changed that break. So what is gene editing? It really is taking a region and changing it to what you want it to be. Yes, I could go into all kinds of structures, crystal structures, show you sequences, but in the end, this is really all it is. All right. So how can we now control that break? How can we make sure that break happens exactly where we want it? Because you don't want to just randomly break your genome and hope that one of the breaks ends up in the place you want changed. Let's say the gene for breast cancer or the gene for colon cancer. We took advantage, we being whole field geneticists, of a set of enzymes called endonucleases. Now, nuclease, that word ace, the ending ace, means break. Nucleases are enzymes that break nucleic acids, such as DNA, which is one of the genetic chemicals in our body. There are so many nucleases. Some start here and eat this way, others start here and eat that way. Some of them start in the middle. The ones that start in the middle are called endonucleases. They're the nucleases that hit in the middle. And those are the ones that were most interesting, because most of the variants that people might want to change through editing are sitting in the middle of chromosomes and not at the end. So now I'm going to mention 3 enzymes that you may want to watch for in the news. The first one are called zinc finger nucleases. Fortunately, we abbreviate that as ZFNs. I'll describe these in a moment. The second endonuclease that is really attracting a lot of attention is a class called transcription activator-like effector endonucleases, abbreviated as TALENs. How do they work? Well, the goal, remember, is to make a break at a specific place that genome sequencing has told you may be responsible for a neurodevelopmental disorder, or a cancer, or a susceptibility to, inability to metabolize codeine fast enough, or too much ability to metabolize codeine. How do they work? Well, you need a scanning system. These are really, I remember when these proteins were discovered, oh my gosh, we would look at them and think, how beautiful is this molecule? It was like it was made to be appreciated. So these, both zinc fingers and talons, have an arrangement of what we call subregions, sometimes 20 of them, sometimes fewer of them. Each one of these subregions of the protein, so this one I'm drawing a cartoon protein with 4 subregions. Each one of those subregions bind a specific sequence. And it turns out that after researchers studied these proteins for a long time, they could pick any sequence they wanted and design a protein to recognize those sequences, because they broke the code. They broke the code of recognition between the protein subregion and a DNA sequence. So then, companies formed for a lot of money. I never was able to use one of these companies, but you would write in and say, you know, I want to make a break at this sequence, and you would send it in, and the company would say, okay, we'll send you the protein in about 3 weeks. And they would sit there and make the protein, send it to you. All right, so here's this protein. Imagine there are various ways in which you can get it into the cell. It sits down and it scans the entire genome. I mean, there are 3 billion bases, but this protein will scan the entire genome. And when it finds the sequence that you've designed it to find, it will sit there. Now, in reality, you make these proteins in pairs. One of the pair binds to one side of the sequence that you want changed, and the other pair binds the other side. In addition to making these proteins recognize their sequence, they are appended to an enzyme that cuts DNA. So really, when I was a graduate student, a lot of what we did was to try and understand how biology worked, understand how the cell worked. It's completely different now. I shouldn't say completely. There's still a lot of people who work, I'm one of them, tries to understand how biology works. But we now understand how these proteins work well enough that they've become what we call our tools. And we have a toolbox with hundreds and thousands of tools. We may not understand how the cell uses these tools, but we know what these tools do. And so now, one of the reasons genetics is moving so quickly is we know how to mix and match these tools to create nanomachines that do exactly what we want. This is a human-made nanomachine. It's designed to recognize a sequence, and then after the sequence is recognized, the molecular scissors that we've attached to this protein cut the DNA. And then the rest is what you've seen before. The DNA is cut. If we introduce into the cell a template that we want, a sequence we want inserted, it'll get inserted. And that is what zinc fingers and talons have been doing. They are really remarkably precise machines. And so zinc fingers and talons gave us an answer to the dangers of random insertion. They gave us targeted edits. Huge celebration for that. But they did not address the low efficiency issue. So there are plenty of gene therapy trials ongoing. A number of them use talons and zinc fingers, but the efficiency is low. So there are other techniques we use to counter or to accommodate their low efficiency. What happened is that in 2010, 2011, this group of individuals studying bacteria, the immune system, how bacteria protect themselves from viruses, discovered this enzyme, which I think many of you have heard of, called Cas9. Cas9 implements the CRISPR protective system for bacteria. And they were studying it primarily, actually, because they were making their company was making yogurt, and they needed to get rid of the bacteria that were invading their yogurt cultures. But as they looked at this system, they realized that the CRISPR-Cas9 machinery had an exquisitely fast, efficient way of searching genomes and finding their target and cutting the target. So, although you probably have heard there's a lot of competition among scientists, I'm going to tell you my personal experience is people are people, they will compete. But 99% of the time, in a conference, people will sit down and figure out how to make things better. So I was just at a conference before I came here, and I'm kind of an old-fashioned geneticist. I work on weird things. And because of what we invented for looking at the genome, I've been slowly moving into a faster-paced field of genetics. And I could feel the intensity in this room. It was really extraordinary. And I know a lot of the players, so I know them, and we're talking. And what I was really struck by was how hard competitive groups worked to put aside their differences to make the field go faster. It's a lot of work. It's not easy to share with your competitors But that is exactly what happened in this room. And I was really, just wanted to share that with you. So then, this group of individuals saw the publication. They worked a lot in mammalian systems and also in gene transformation. And so I would say the 7 together put together what we have now as the CRISPR-Cas9 revolution. Now, this is my conflict of interest slide. As already mentioned, George is my husband. People often ask me, did I fall in love with him because he was so smart? And I can honestly tell you, that when I met in graduate school, what really struck me about him is he's so nice. He was just the nicest person I had ever met. So I have no conflict. I know whenever I call this a conflict of interest slide, I feel like just not doesn't really quite express the fact I have no conflict with George. But it's a family. Turns out Jennifer Dowden was a graduate student in our graduate program many years ago, and I can tell you, the second she walked on campus, all the faculty knew she was brilliant. So we've known Jennifer for a long time, and I also need to acknowledge her because she gave us the $100,000 from her Gardner Award to support PG Ed. And I just got an email today saying she has contributed another $100,000 because she is so anxious about how this CRISPR-Cas9 revolution is going to go, and she wants to make sure that there's public engagement. Feng Zhang is also like family. He was George's postdoc, and so I've known him since he was in George's lab. I did not know Emmanuelle Charpentier, but now I've met her. And in spite of what you might have read in the literature and the media about the patent wars between a couple of these individuals, I want to tell you that scientists mostly talk science. So when you get them in a room, they're talking about their experiments and what to do next. And this is, I want to bring this up, and I emphasize it a bit because this is also a piece I think is something the public may want to weigh in on in terms of the ethics of patenting and what we do with genetics. Okay. So, CRISPR. CRISPR does not look like this. These are expensive proteins to make. They work beautifully. In fact, they are more precise than CRISPR, but they're very expensive, making this an elitist sort of research. Most scientists cannot afford them. What is so cool about CRISPR-Cas9 is this searching mechanism is replaced by an RNA mechanism. RNA is really easy to make. It's so easy, you can make 100,000 of them and not hurt your budget too much. So this is what made CRISPR-Cas9 suddenly possible for just about every single lab, even for do-it-yourselves. The scissors come in on the protein called Cas9, but it works exactly the same way. It cuts, and then you can put in a template DNA and allow you to make changes. So what CRISPR-Cas9 has done is to give us a targeted system and to address the low efficiency. It's not, again, currently everything's getting better. It's about 4 times as efficient as zinc fingers and talons. It is not quite as error-proof as zinc fingers and talons. It is far less expensive. It is hugely efficient. It used to be you would plate out a cell of about 100,000 cells and maybe you would get a few transformations. Now you can plate out a small set and expect to get many, many transformations. So graduate student projects go much more quickly. What has it done for us? One, it's allowed us to test disease variants in model organisms much more quickly. What would have taken 5 to 10 years to do, people can now do sometimes in 3 to 4 months, greatly reducing the cost of genetic research. It allows us to tailor our experimental systems very quickly. Again, speed, less cost. Most of our work is supported by taxpayers. This is something that George has been involved in. It's been very exciting. Transplants. Most people do not receive the transplant organs that they need. They don't receive them in time. Many years ago, people noted that pigs have organs about the right size for transplants into humans. And some efforts were begun to think about using pig organs as donors for humans in need. But it was also discovered that pigs carry a retrovirus that can jump out of the pig cells and into human cells, making it not a very attractive system. So what George's lab did, in particular Lu Hanyang did, was to take the pig genome and use CRISPR and get rid of all 62 retroviruses in the pig genome, making it a safer genome. And they made some other changes to make them better tissues for transplanting. And I can tell you, as of a few weeks ago, a little piglet was born. Actually, many piglets were born. They are retrovirus-free, and soon there will be trials beginning to see how well their organs will do in terms of compatibility for transplants. It is not a universal solution. There are ethnic groups that cannot use or do not want to use pig-derived materials. I was looking at some articles recently that said that for some of them, for the Muslim communities, it's a very big issue. For the Jewish community, it's a big issue. But the imams and the rabbis, some of them have said that under dire circumstances, with no other choice, pig materials are possible. But I think efforts are now ongoing to try and see if they can use sheep to make this a more accessible and uniform process. And this is Lu Hanyang, brilliant student, who, when she was a graduate student, was actually told was possibly not good material for research scientists. And it was because of, we think, her ability to express herself. So George worked with her very hard, and she is now an amazing rising star. She is unbeatable, so remember that name. Ecological vector control, gene drives. This is something that also needs a lot of discussion in the public. Malaria is a very good example to understand this situation. So malaria is caused by a bug transmitted by mosquitoes to humans. Millions of people are affected, die from malaria. The thought was, well, we can make mosquitoes that are resistant, that will not pick up the malaria bug. It's a little easier to think about altering a mosquito than altering a human. If the mosquito cannot pick up the malaria bug or kills the malaria bug when it's in it, then humans will be protected. But it's a really, you can tell. Let's say the pink mosquito is the one that is resistant to malaria. In the regular mating scheme, it is not going to take over the population. But with a CRISPR-based technology called gene drives, if you put this technology into mosquitoes, rapidly all mosquitoes will become resistant to malaria. It's been in trial, and they're doing a similar thing, they're thinking about doing it for Lyme disease. But it requires the consent of the public, the understanding of the public that this could change the ecosystem for an insect. And mosquitoes actually are a pollinator for some. It's a problem because some people get malaria and die, and other people need to maintain their fields. So it's something for us all to think about. Okay, therapeutic editing of the soma. CRISPR works so efficiently that you can take blood from a person and change most of the T cells in the blood and put it back in the individual. So is it scary? It is a little scary, but there are currently 2,200, actually well over 2,200 now, gene therapy trials already ongoing, inserting whole genes, using, in some cases, using viruses that insert the genes anywhere. So there's a concern that we need to move more quickly using CRISPR, because CRISPR is more targeted. In 2009, people used zinc fingers and addressed HIV by knocking out the CCR5 mutation. They used talents and were able to work with 2 girls who had leukemia, and I think that was successful. In 2016, a published paper came from China where someone used CRISPR outside the body, harvesting cells, CRISPRing it, putting it back in the cell to treat lung cancer in China. And just recently, More trials have started, many of them in China, but also elsewhere in Europe and the United States, using CRISPR to address breast, esophageal, kidney, prostate, bladder, colorectal, HPV-induced, Epstein-Barr-induced cancer. And there is tremendous excitement that these technologies are going to be able to help address these cancers. Companies have arisen. This is Editas. These are some of the diseases they're looking at. One causes blindness, there's sickle cell. This is just the beginning of what may be possible. These are most of the CRISPR-based companies that we have out there. The ones in yellow are the ones that are actually doing human therapeutic research. And then, of course, germline therapy. This is the one where you actually will change the germline of individuals. In December of 2015, there was an international meeting to discuss the ethical use of CRISPR for gene editing. And the consensus was, let's think about this. In February 2017, just a little over a year, the NAS decided that under special circumstances, absence of reasonable alternatives, restriction to genes associated with severe disease, credible data on risk and benefits, rigorous oversight, long-term follow-up, and public input, that they would accept studies that might involve germline editing. And then just this August, this gentleman in Oregon improved the CRISPR technology, demonstrating that it should be possible to alter human embryos and therefore germline using CRISPR. So this is why we're here, because we're really on the cusp of a very critical time, exciting and also concerning. Okay, I think I have 2.5 minutes left. These are some of the genes. People talk about enhancement. These are some of the genes that are called protective genotypes. They protect against HIV, stomach flu. I don't have that allele. Stronger muscles and sensitive to pain. These are things we can talk about. And you know what? No, I'm not. There are some really exciting things about genetics that I just wanted to tell you, even I think as exciting as CRISPR. This is a study, and I can talk more about it during the discussion time, where people have evidence that the experience of a parent, this was done in rodents, can be remembered in the progeny. So this is a non-DNA based form of inheritance. And I bring this up just to let you know that CRISPR is just one of the amazing things going on in genetics right now. Here's a case where people have found a protein in blood that can reverse aging of muscles, aging of neurons, and people are rapidly trying to identify what this protein is in an effort to slow down aging or even reverse aging. It's even being used in a trial for some individuals with Alzheimer's. We can now, in rats, take a somatic cell, like from your skin or some other part of your body, and in a Petri plate, get those cells to generate egg and sperm. So you no longer have to use your gonads, maybe, at least for rats. And they've made mice. Now, I have to tell you, these mice are not eggs made in a dish and sperm made in a dish. These are mice from sperm made in a dish with a regular egg, or egg made in a dish with a regular sperm. But you know where this is going. And the good thing is, they're saying we should not do this too much more unless we have public input, which is another reason I wanted to mention this to you. Finally, nanorobots. This is fun. DNA is a material, science. We can fold it into any structure we want. This is a molecule. We call it a nanorobot. It is a basket where the basket is open. It's got some sensors in yellow. It can sense a chemotherapeutic drug. When it senses enough chemotherapeutic drugs, it triggers, it closes. The corners of the basket can sense the difference between a cancer cell and a normal cell. And so the hope is that this will allow us to target deadly chemotherapeutic drugs only to cancer cells. Because otherwise, chemotherapeutic drugs are a race between killing the person or saving the person. And finally, this is a good way to end. DNA can also be used for DNA storage. This is my husband worked with Technicolor and encoded this first amazing movie, this is a landmark movie in Hollywood, into DNA, and then they took that DNA and decoded it into a movie, which is what you're seeing. So the movie you're seeing now came from DNA stored information from the movie. So there are many, many things that can be done with genetics in your body and outside your body. And I think that I will stop there. Thank you very much.
Possible that no matter how much we may discuss the ethical consideration, there will be few actors outside that circle. They'll go ahead and do it and make these discussions not very relevant.
So, do I think that no matter how much we discuss it, will there be some rogue do-it-yourselfers maybe that do something? I think, given how human beings are, probably. And so one might say, why discuss it at all? I think we are all, one way to think about it maybe, is that we are all caretakers of each other. In the past, some terrible transgressions were made in the name of genetics by geneticists, politicians, sociologists. It led to the eugenics movement. People being sterilized, the Tuskegee event, the Holocaust. Terrible things were done. And it may be, I think many of us feel, many geneticists feel, that it was because there was not enough public awareness of what was happening. So people did not know what was happening, and they weren't able to comment, and so terrible things happened. And this time, my colleagues and I want to make sure we don't make that mistake again.
Thank you very much. Lovely talk. Can you comment on the ecological effects of something like a mosquito being edited because it is consumed by other animals?
All right, so the ecological effect of a mosquito being edited, will it affect the organisms that normally eat mosquitoes? The hope is not, because not all mosquitoes carry malaria. If malaria were an essential part of the mosquito life cycle, that would be a problem. But it's not, so hopefully not. There are negotiations ongoing now to find islands or populations where this can be tested. It is definitely a scare. You know, when everything turned purple, it made everybody, it makes you worried. So thank you for asking this question. I forgot to mention that the CRISPR people went back into the lab, and they've now made more complicated CRISPR gene drives that have limited effect. So they eventually stop working or are reversible. It's not a fail-safe situation yet. Right, so the patent issue. So I used to be in exactly that camp, which is odd because I am married to George, who has all these patents. And we would talk about this all the time. I'm telling you this so you might think, oh, she's completely bought into it. But I actually hold a number of patents myself, because one day a colleague of mine came up and said, and this is a bit of an exaggeration, but this is what she said to me. She said, imagine you have the cure for cancer, and you don't, for every cancer, and you don't patent it. She goes, no company, no person, no university has the funds to take it through all the clinical trials and go into debt. No one would do that unless they had the chance to make up those funds. And that made me start to think about it. But the other thing I learned about patents is when you hold a patent, you can control how it's used. So for example, just the other day, I've been talking with some companies, and there's one that just wants to enable this technology and will allow researchers and hospitals to use it at cost. So I talked to the patent group, and they said, you get to choose. You get to choose where it goes. So hopefully, the more public comment comes into scientists and business people, the harder it will be for them to make a decision that is going to be not beneficial to the public. And I think public voice carries tremendous weight with scientists. So it could happen, but I'm hoping not.